Welcome. The Passive Sampling Technology Update team is proud to present an overview of the regenerated cellulose dialysis membrane sampler technology. This quick video will provide a high-level overview of the device, as well as instructions on how to use, deploy, and retrieve the device. For more information on this and other passive sampling technologies, please visit the Passive Sampling Technology Update Guidance document. The Regenerated Cellulose Dialysis Membrane or RCDM sampler is an equilibration-type passive sampler. RCDMs operate using the principles of molecular diffusion across the regenerated cellulose dialysis membrane, filled with deionized water. The RCDM sampler is primarily used to sample groundwater, but can also sample surface water and sediment-poor water. The device's regenerated cellulose membrane allows for the equilibration of many types of chemicals to reach equilibrium over time, including cations and anions, nutrients, dissolved hydrocarbon gases, some volatile, semi-volatile, explosive, and PFAS. RCDM samplers use molecular diffusion across the semi-permeable membrane to sample the surrounding medium. Generally, chemicals tend to migrate from an area of high concentration to an area of low concentration until equilibrium is achieved. The concentration gradient between chemicals in the target aqueous media and the interior of the membrane allows for this process to occur. The rate at which equilibrium is attained is determined by a number of factors, including the magnitude of the concentration gradient across the membrane, the pore size of the membrane, the size of the chemical species, the temperature of the water, and the hydrophobic or hydrophilic nature of both the chemical species and the membrane. A fully assembled RCDM sampler is not commercially available, but the individual materials can be purchased and the device can be constructed prior to their deployment. The main parts of the RCDM sampler are a pre-cleaned regenerated cellulose membrane, a disposable plastic clip or zip tie to close the top of the membrane, a disposable PVC valve and a plastic clamp connected at the bottom of the membrane for sample transfer, a protective mesh bag to protect the membrane from abrasion during deployment and retrieval, and a line to suspend or fix the device in the sampling media. The RCDM samplers may be constructed with PVC pipe segments installed into the ends of the mesh to maintain sample volume and sampler integrity in low ionic water. For high ionic water, a rigid low-density polyethylene mesh or perforated PVC pipe can be inserted inside the membrane before the sampling valve. To construct the RCDM sampler, cut the membrane into lengths long enough to enclose the desired volume that will be needed for all analyses. The maximum length should not exceed the monitoring well screen or bedrock sampling interval, which is generally 5 feet in length. Next, clean the membrane with deionized water to remove any preservative used to ship the membrane. Clamp the top of the sampler with zip tie or plastic clamp and attach a PVC valve at the bottom. Encase the sampler with a plastic mesh to protect the sampler during deployment and retrieval. Next, make sure the appropriate pipe segments are installed depending on the ionic state of the sampling medium. PVC sections can be used for low ionic water, and a rigid low-density polyethylene or perforated PVC pipe can be inserted inside the membrane for the PVC valve is attached and sealed. Last, fill the RCDM sampler with deionized water. The sampler should be constructed within a few weeks of deployment and must be kept immersed in deionized water between construction and deployment. Some methods to keep the sampler wetted include keeping the samplers in a low-density polyethylene sleeve knotted at one end, filled with deionized water, and then clamped at the other end. Alternatively, the samplers can be submerged in a clean plastic bucket or PVC tube filled with deionized water. When ready to deploy the RCDM sampler, make sure the sampler is still filled with deionized water, and the membrane and the mesh are in good condition. For groundwater monitoring wells, measure the depth to groundwater, and ensure the entire device will remain below the static water level. Adjust the length of the supporting line based on the sampling interval within the well. If sampling surface water or sediment-poor water, make sure the sampler is fully submerged and will remain submerged during the deployment period. When ready to retrieve and collect samples from the RCDM device, ensure the proper equilibration time is achieved. Remove the sampler from the sampling medium. Remember, use caution when handling the device as to not perforate the membrane. Next, cut any protective mesh to access the sampling valve and rinse the valve with deionized water if any sediment has accumulated. An extension tube can be attached to the valve to direct the sample into the laboratory-supplied bottles. Some of the main advantages of the RCDM sampler include: RCDM samplers provide a sample of dissolved chemicals, keeping out suspended particles. RCDM samplers have been lab and field tested for a wide range of commonly sampled organic and inorganic chemicals. RCDM sampler volume is dependent on diameter and length of sampler. The volume contained can be easily increased or decreased during construction, unlike some other equilibrium samplers that are volume limited. Some limitations of the RCDM sampler include: RCDM sampling devices are not commercially available, so they must be constructed by the user, and this requires some training. RCDM samplers must be kept hydrated in DI water between the time of construction and time of deployment to maintain the permeability, flexibility, and strength of the membrane. RCDM samplers can biodegrade within 4 weeks, depending on groundwater temperatures and bacterial populations, resulting in perforations and partial to total sample loss. Some important takeaways about the RCDM sampler include: RCDM samplers can be used to measure a wide variety of chemicals and can be custom-built to achieve sample volume needs. Fully constructed samplers are not available from commercial vendors, and samplers must be carefully maintained during construction and shipment. Samplers may biodegrade within 4 weeks, so the time between deployment and retrieval must be considered carefully to achieve equilibrium conditions without potentially compromising the device. Remember, you can learn more about the RCDM sampler and others in the ITRC Passive Sampling Update online guidance document.